Okay, so the last time we met, we were talking about our viruses. And this is definitely an area you want to start studying now. Again, just like you did your prokaryotes and eukaryotes, get your groupings figured out and start studying with your groupings. I believe the last one we talked about was the, let's see, which one was it? Was the SARS one, correct? Is that the last one? Yeah. So SARS, as I mentioned, was an outbreak in China in 2004. And now we also have COVID that's here, so SARS and COVID. COVID and SARS are closely related. SARS stands for sudden acute respiratory syndrome. It, as I mentioned before, has very similar symptoms to COVID. The only thing that's different was that it was much more aggressive and the symptoms showed up within 24 to 48 hours. COVID was harder for us because you had a 2-week wait sometimes, and so people could be spreading it and not know. And then this one's our Ebola virus. Actually, I think it was this one we left off on, huh? The Ebola virus is one that we've mentioned before, that if you survive and if you live, did I skip one? Okay. If you survive and if you live, you can have, unfortunately, long-term effects. Ebola has a tendency to cause joint damage because of scar tissue. A lot of Ebola survivors have to have hip replacements when they're in their 40s because of scar tissue. And then this is a picture of an eye of a person who had Ebola. And so, as you can see, there's the optic nerve, and then there's this green coloration. That's all scar tissue from the virus that was left behind. And that scar tissue doesn't go away, so you basically get like an Ebola cataract. It's something that will affect your vision for the rest of your life. What's really crazy is when somebody has active Ebola, if you do an eye scan, you can see the virus swimming inside of it. Oh, that's crazy. Isn't that crazy? It's like anything that's in your eyes moving just gives me the heebie-jeebies. You know, like, have you guys ever seen that show, The Monsters Inside Me? Yes. You knew where I was going. Estefani's like, yeah. Yeah, Monsters Inside Me. It's like you see like worms swimming through the eyes. Oh, it's like that's, as a microbiologist, that's the only thing that grosses me out. Things shouldn't be swimming in your eyes, you know? So it's just like, oh, okay. The next group we're going to talk about is our ortho group, Orthomyxoviridae. And this one is one that we're really familiar with because it is influenza. And you might have wondered, what is the deal with flu? Why do we have to get shots every year? Why is it, you know, some years are worse than others? And so the deal with influenza is that there are a lot of different strains and variations out there. We mentioned surface proteins like H1N1, things like that. That affects how well the virus can attach. It also affects how similar the virus is to a previous strain. And so if our body sees one that it's seen before, our immune system can fight it off quickly. If it's a strain we've never seen before, that's when it can be really dangerous. So COVID, when we had our outbreak of COVID, a lot of researchers were mentioning Spanish flu. Did you guys hear that on the news, Spanish flu? So Spanish flu was one that was around 1918 to 1920, and it was a very contagious flu strain, and it started in Spain, which is why it was called Spanish flu. Honestly, it probably was not really started there. They were just the first country to report it. And so what happened with Spanish flu is it was a particular strain that we had never been exposed to before. And it just swept across the country because everybody was basically susceptible, kind of like what we saw with COVID. The difference with Spanish flu is it also led to pneumonia. And so people were basically dying of drowning in their own fluids. So their lungs would get so full of fluids they couldn't breathe. And it had a very high death rate. And today, that same strain of influenza has not circulated for decades. Now, at the CDC in 2015, they had a group of researchers that were studying it, and they meant to grow one strain of flu, and they accidentally grew the 1920 strain of Spanish flu. And it ended up getting into the ventilation system because they weren't using the same precautions. They were thinking it was a class 2, and Spanish flu is actually a class 3. So they didn't have the same precautions, and everybody who worked with it got sick. It went into the ventilation, and the entire 3rd floor of the CDC got exposed. And so they were all hospitalized. Luckily, nowadays, we have something that we didn't have in the 20s, which is ventilators. And so we were able to have people go into induced comas to help them get through. But it was the same thing that we see with COVID. A few survived. Most of them survived. A few died. Did you guys see that in the news? Yeah, they never talk about that. Isn't that funny? There's a lot of little mix-ups that happen, and you never hear about them. Another one that happened was there was a researcher at CDC who was sampling COVID patients who stopped at McDonald's to get a coffee and took in his tray of samples because he couldn't leave them in the truck, put his tray down at the McDonald's, ordered his drink, left the tray there. So then later on they get a phone call and McDonald's is calling this research company that's on the side. They were calling the 1-800 number on the side going, do you want your sample? There's some stuff here on the counter. We don't know what it is. Like they were like not sure what to do. I mean, so again, you don't hear that in the news, right? So it's always funny to kind of read about what's going on sometimes in the science world because it's like whispered about in the medical community. It's always interesting. So influenza vaccines. Have you noticed that some years they work better than others?
Yes.
Yes. What's the deal with that? Well, the deal with that is we have to guesstimate what flu strain is going to be in our area during winter. And the way we do that is by looking at other countries that are having their winter at that time. So we'll look at Australia, we'll look at other countries in the springtime and say, okay, what's circulating there? And then we'll inoculate our eggs, we'll grow the flu vaccines based on what's in circulation. And sometimes it stays the same, and sometimes the strain mutates or changes, and our vaccine is not effective at all because we grew for the wrong thing. Yeah.
Do epidemiologists gather this data like year round?
Yes. So they are constantly gathering data of who has flu, where flu is, what outbreak it is, and what strain it is. So they monitor all of this. There's a report from the CDC, and we'll talk about in the next chapter, called Morbidity and Mortality. Morbidity is the number of illnesses or cases they have. They track that. And so they just keep a constant influx of data. Great question. And so sometimes the vaccine works great because we get the right strains, and then sometimes it's not effective at all because the strain changed, and the one that's here is not the one that we prepared for. The problem is the vaccines take time, so we can't really just flip on the fly. We have to just kind of go with it and hope that it works out. The last one we're going to talk about is the HIV virus. And as I mentioned, this is a really oddball one because it does a lot of extra steps than what it really needs to do. It's a retrovirus. So it comes in as an RNA, and the reason it's called retrovirus is because it goes backwards from the RNA to make a copy of DNA. So it goes RNA to cDNA, and then the cDNA makes another copy of RNA, and then from there we go into the ribosome. So that was on the previous slide we had about HIV. So we just do all these extra steps. The nice thing is because when this virus has to make its cDNA, it needs to use an enzyme called reverse transcriptase, we can wipe that enzyme out, that reverse transcriptase. And that stops it from being able to replicate. So if you're exposed to HIV, we can't get rid of it, but we can stop it from replicating. So it'll always still be within you, but at least it's not replicating. So that's the good thing. And what that means, it's within you because every time that cell divides, the virus still divides inside of it. But it's not able to make a whole bunch of offspring. Now, the reason we worry about HIV is because of what it attacks. And it targets and attacks our T cells. And these are part of our immune system. And so what's going to happen with this then is when they target our immune system, they're taking out one of our first responders that is going to react anytime we get exposed to any kind of virus or bacterium. So it's an odd illness because technically nobody dies of HIV. HIV progresses and eventually it'll wipe out enough T cells to where it wipes out your immune system. And that's when you move to AIDS, acquired immunodeficiency syndrome. That's what AIDS is, acquired immunodeficiency syndrome. When you move to AIDS, that means you have hardly any T cells left at all. And again, AIDS doesn't kill you. What kills you is any virus or bacteria you're exposed to. So people will die of a common like Pseudomonas ear infection. They'll die of thrush with yeast. I mean, things that should not kill you. So as you can imagine, when HIV first showed up in the 80s, it was quite baffling because we had people dying, but they were all dying of different illnesses. It's kind of like what we see with long-term COVID. You know, it's hard to say what's being caused by COVID and what's being caused by the body's inflammatory response. There's kind of a point where we're not really sure how to separate the two. And that's what happens with AIDS. It's like, well, AIDS technically didn't kill you. It was whatever you were exposed to afterwards. But the bottom line is the same. It's still wiping out our T cells. The virus, if it's unchecked, can infect about 10 billion T cells every day. Our body can only replace 1.8 billion. That's the way we're set up. So as you can imagine, it has a huge advantage on us. So it's killing much faster than what we're able to replicate or produce. And this is why our T cells get, killed off so fast. Again, T cells are first responders that help with targeting and destroying bacteria. So eventually what happens then is that the body's struggles start to reach a point where those T cells just aren't present enough. And again, we're unable to fight anything off. And the way you can acquire AIDS is usually through blood contact, through intercourse, or through like organ transplants, blood transfusions, something that has a blood or tissue contact. That's why it's considered to be a class 2 when you're studying it, because when you're working with it on the bench, as long as you don't have any open wounds or anything, then you're completely fine. But it is one of the things you have to be really careful with if you're doing blood draws on somebody who's HIV positive, that you don't accidentally stick yourself, because then that would be a scary situation. Okay, so that gets us to the end of our viruses. And so now we're going to jump into epidemiology. And this is a really fun chapter because this one's going to be talking about how we study all of these diseases, how we came about studying them, how we learned about them, and then what we're doing today to try to keep people healthy, which is really nice. So with epidemiology, when we talk about that term, it really can break down to make a lot of sense. So epi means on or upon. So it's a virus or a bacterium that is living upon or in our population. Demos means people or population, so it's saying it's living with us. And so epidemiology is where we're studying how diseases are occurring, how they're spreading, and what their impact is. And as you can imagine, epidemiologists are tired right now. They have been working around the clock with COVID. Part of the problem with COVID is it was so new. They were trying to, the best analogy I heard was build a plane and fly it at the same time. And that's, I think all of us were kind of struggling with stuff. And it's the same idea. They were trying to share information about COVID as they learned information, and that kind of backfired on them because sometimes they would say something and then find out that wasn't true and have to recall that statement. And so people started losing confidence in them. Yeah, Misha.
I have a question.
Yeah.
So would it have made, I kind of know the answer, would it have made a huge difference if we were able to know more when it was first popped up in China, which a lot of people don't like to even address, but
yes. If we had known more and had more warning, it would have been a huge difference. We know for sure, there's a lot of debate on where it came from, whether it was a class 4 that escaped, which is what most microbiologists believe, or whether it was, something from the environment. We know for sure that China knew they had a problem back in October.
And they just didn't want to say
it. And they didn't announce. They were hoping to keep it confined. And the reason we know that, hospitals and medical companies, we buy our lab coats, our gloves, all of our medical equipment, most of it from China. And in October, all of a sudden there were back orders. They weren't shipping things out, they were keeping them in the country, which means they needed them. And then all of a sudden there wasn't as much travel. And scientists who were supposed to attend conferences weren't coming. And so there was kind of a question of is something going on in China? So people were kind of asking back around November, December if things were okay. We also saw a huge increase in the purchase of urns and coffins. So we know they knew they had a problem before they announced. Regardless of where it came from, it was definitely kept touched.
Normally when there is a disease or something that pops up in another country, is it the WHO that's allowed to go in and test it or
World Health Organization and CDC. CDC actually has representation in all of the countries, but here's the deal. Our Wuhan person had retired in July of that year, and they had not replaced them. So we did not have a field agent, and that killed us. And not having a field agent means that we couldn't have anybody go to the marketplace before they bleached it. We couldn't have anybody go to where the first diseases came out because they bleached everything. And so there was nothing that we could recover at that point in time because we didn't have an agent in the field. And by the time it was announced, we couldn't travel into the country because it was closed. So that was like a 6-month cleanup job. So we couldn't get any data at that point. If we had somebody stationed there like normal, it would have been okay. But they were actually thinking about getting rid of it because of budget cuts. So they had to replace that position. So it was like, a domino chain. Everything that could have gone wrong did. And so unfortunately, we just didn't get the kind of advanced warning that we wanted. We suspect now that we probably had COVID circulating in December here. There were a lot of people that had the flu that wasn't going away, right? And so we suspect that it was probably here. What about last year? Do you guys really think there wasn't flu last year? There was flu last year. It was being misdiagnosed as COVID because at that point in time, it was just easier if you had respiratory syndrome to treat you like a COVID patient. And most people, that was just rest, trying to get as much drinks as water, hydration, that kind of thing. But we had a lot of misclassification. The flu did not just vanish. The flu was always with us. One of the things that helped with our numbers is we were in more isolation. So the numbers did go down, but we still had it circulating. Adrian.
I was going to ask if COVID is the only known circulating virus or pathogen that causes loss of taste and smell.
Yes. So usually with a cold, when you're stuffed up, you can get a loss of taste and smell because when you're congested, your nasal passages can't process that smell. But COVID is weird because your nasal passages are completely clear, but you still lose taste and smell. And so that's like one of the biggest signs. And one of the things that they've been having lately is they have lost that as a symptom. So as the strain is mutated, they're saying that sometimes people are still keeping their sense of taste and smell, which is kind of interesting. So it's kind of a mutation that comes and goes. But one of my friends had COVID, and she said that she knew she had COVID because she went to wash her clothes, and the detergent she uses is lavender scented, and she couldn't smell it. And she said she opened the box and she was waiting for the flower scent, and there was no flower scent. And she went down, was smelling the box. She's like, I had my face in the detergent and I couldn't smell anything. And she goes, and I walked in the house and there was coffee going and I couldn't smell it. And I was like, oh no, I knew what it was. That was the only symptom she had, was the loss of smell and taste. She never got fever, never got upset stomach, nothing. That was her only symptom. So definitely don't ignore that if you have that. So she was very fortunate that she got it. Good questions. Any other questions?
Do you think that with the flu season coming and the holidays approaching, there'll be somewhat of a spike of COVID again, for sure?
Definitely. So one thing I've noticed with COVID, it seems to be doing a cycle.
Yeah.
We've been talking about that. So right now we're in the downstage. We're going to have an upstage here pretty soon. Probably around at the end of November, right around Thanksgiving.
Yeah.
People are going to be traveling more. And so we're going to see an uptick. But also, it's just naturally, the virus seems like it mutates and then comes back, and then it mutates and then comes back. So we see this kind of cyclic cycle. It's like every few months we have a surge. So we're kind of waiting for the next surge. It'll probably be around November, December. I do expect it to be increased. I also expect this flu season to be increased because we've all been isolated. And now we're going out there and our immune systems have forgotten the flu. So when they see it again, they're going to be like, oh, what's this? So definitely if you can do a flu shot, do that. It's effective for most of the strains, but definitely if you travel, even if you're vaccinated, wear your mask. Definitely wear your mask.
This flu actually lingers for a lot longer than last time.
Yes, because our
My
immune
kids
systems
got it.
are lower. We have been protected, and so it's kind of like, remember I told you about my story about my cousin who was the Purell queen? Did I tell you guys that story? Yeah, Purell queen, and then she sent her kids to school and they all got sick.
Oh, yeah.
Yeah, it's the same thing. We have been isolated for over a year and a half, and we're slowly getting back. But when we start traveling, all of a sudden we're going to be exposed in mass, and everything's going to be hitting us harder. Yeah.
I have a question. So, I don't know, taking this class, we learn a lot about vaccines and things like that. So, why do you think there's a lot of people in the health field that are, so against it, and then it's just leaving that, picture for everyone else that doesn't really even believe in COVID? And, why wouldn't people in the healthcare, if they've learned about all of this, why wouldn't they understand it?
Let me ask you this. Do you guys know anybody in the health industry who smokes?
Yes.
Do you know anybody in the health industry who's obese?
Yeah.
Why? They know of the effects. It's just, it's a personal choice. So with the COVID, originally a lot of people were saying they didn't want the vaccine because there wasn't enough research on it. But we reached the point where there is really good research. And now they're saying they don't want it because it's mandatory. It's more like a principle thing. It's like, well, you have a driver's license because it's mandatory. So are you going to turn that in? So I would just caution you to always do your own research. Just because somebody else is doing it, even with getting a vaccine, just because somebody else is getting a vaccine, always do your own research and, decide for yourself. But I'm always cautious with following what healthcare people are doing, just because, a lot of people make decisions that just don't really make sense to me. Even though they're people that I would expect to have a smarter approach, I've just kind of learned everybody makes their own choices. But that's a great question. So a lot of it is also fear. If you go online and look at anti-vaxxers, they have amazing videos. I have to tell you, I'm a mom too. And I was doing my research, as a microbiologist and as a mom when my kids were little. And I gotta tell you, it's scary to take your kid to get a vaccination, even with all of my background and knowing what things do. I mean, you read these reports online, and it's like my kid's head fell off, it rolled across the room, we duct taped it, it's okay, but they've never been the same. They walk a little wonky now, you're like, what? You're like reading this stuff, and it's terrifying. The reason, too, there's so much bad stuff on the internet, nobody ever goes home and types up and says, had a vaccine, things are great. You're not going to post on that. Yeah, you're not going to post on things that go, okay, it's kind of like a restaurant review. People tend to post the bad things more than the good. So we always have to kind of take things with a grain of salt. So, but they're very well written. I mean, they really do a good job of kind of laying
pretty
theory.
convincing. And then
I think some people are just over dramatic too. Some people just can tell stories.
My daughter is the most dramatic child in the world. When she gets a shot, she's on the floor. Ah! It's really, you're not stepping over. Do you have chalk to do a chalk outline? She's gone. We lost her, obviously. So yeah, exactly. And then, sometimes there is swelling or redness at the site, and people are like, oh my gosh, I'm dying. It's like, no, that's your antibodies. That's okay. You're okay.
I think people forget, though, years ago when there was no social media, I'm totally going to age myself, but you didn't hear about all of these side effects and different things. And sometimes those are already told to you when you're signing for your kids to get this and this and this could happen. It could be, people just forget that, and they didn't realize, and they walk in a lot of fear.
Right.
Most of the students in here are already trying to go into their LVN. I've been at LVN for 20 years, and I've never seen so many people divided with the whole, I didn't even know people were anti-vaxxers until all of a sudden we're on Facebook and they're talking about, I don't want to get this. And it's well, are you really afraid of the vaccine, or is it just, I don't want to do it because they're telling me?
Right, and the skepticism is there. And I think that's, it's kind of like when we were kids. If we were told to do something, we might be, that's fine.
So, my opinion on it is have ever in the history of viruses or diseases like this, has it ever been such a political debate? Because I feel like we've gone from being a health concern with COVID to more of a political concern.
Correct.
So it's not,
Correct.
I understand it's weird, but I think it's more of choosing a side for sure. And has it ever been like that in history before?
No.
And you didn't have social media. I remember when, Tdap or DTaP, when that was the whole whooping cough vaccine became mandatory for students at a certain age. Nobody flipped out this bad, but parents did get upset, and there was a small group of anti-vax parents, but not like this.
Well, it's the same thing with seatbelt mandates, right? We had seatbelt mandates come through, and helmet laws for motorcyclists. Do people get mad? Yeah, they hated it. But when you look around, people are wearing seatbelts and they have their helmets on. It's a safety concern. So it's just, it's something that you have to choose your stance. But it is, I get both sides, but it is hard. It's hard because also this decision impacts others too, and so it's a hard decision.
My cousins were talking about this yesterday. One of them had the argument that if they get the COVID vaccine, it's only going to be working to fight off that, but they can still get the flu or something like that, because we're still saying trying to keep with COVID, not the other stuff.
Right. So every virus is different. So if I get a COVID vaccine, it's specific for COVID. If I get a flu vaccine, it's specific for the flu. So that's why we're recommending both, is because of the fact that each virus is different. I can't target them the same because they're different shapes, different proteins, totally different. Yeah, exactly. It's like comparing a chocolate chip cookie and a peanut butter cookie. They're both still cookies, but they taste very different. Right? See, I always have to do food analogies. Yeah, in my head, that's where I go. Any other questions?
Sorry,
Good conversation. No, it's
Opened
good
up Pandora's
conversation.
box.
I love.
questions.
No, I love that. I love that we have these conversations. Yeah. What's up?
My cousin is like the biggest anti-vaxxer I've ever met in my life, and talking to her, it's like actually scary because she's so convincing that I have to like remember that that's not true. But she said this specific thing that was in the vaccine that caused infertility. She said like a compound, like I can't even tell you what it was, but it was like a 3-letter compound. And she's like, that's in it, and if you look it up, it will tell you that it has that. But I've heard that's like not
Haven't they
true.
done research on it?
Yeah.
Yeah, they've done research, and there's not any correlation between infertility. And then also we have to remember how much.
Yeah.
So if you have something affecting you, so this goes back to like our Diet Coke. Diet Coke has what kind of sugar in it? Everybody know?
I don't even know, but
It's a saccharine sugar. It's like a fake sugar. And there was a study that came out that said if you drink too much Diet Coke, you're going to get cancer and die. Aspartame. Yeah, that's the other one. Okay. So there was like this big study that came out. And they said, we have proof. We have, rats, we gave them aspartame and they died. And so everybody was like, oh, we're not touching it, But then they looked at the study. And for the size of the rat versus the size of a human, you would have to be drinking, I think they said it was like, I have to remember my numbers here. I think it was like 20 cases of Diet Coke a day. I don't know anybody that drinks 20 cases. That's 24 cans times 20. That's a whole lot of diet. It's like you wake up in the morning, you're like, hold on, pop your tag, you're like, hold on. I mean, how? You'd be living your life in the bathroom, right? So it's that thing where, again, we always have to look at the study and we always have to compare it. So yes, it can cause cancer in a huge amount. And same with that chemical, for the amount that's in the vaccine, they're not finding any correlation. But is there some truth behind that? Well, sure. If you get exposed to like a truckload, then yeah, it probably will cause a mutation. Pretty much anything will. If you drink too much coffee, it can cause mutations. I don't want to give up my coffee, it's like, I can't imagine that. But the idea is, you're right, and that's kind of what happens is we get a kernel of truth, and then we build on it. Yeah, and it's exhausting. Like, I don't even try to fight with people anymore. I'm just like, I'm not going there because I just don't have the energy. I'm like, I'm old and I'm tired.
And they will do that. They'll take it and like spiral and
just keep going. Yeah, it's like I
stop arguing with people.
I do have to say, I admire the passion that anti-vaxxers have. I love that passion, because that takes a lot of work, and it's a lot of passion that's there. So, I do admire that. Okay, any other questions? Okay. And as things come up, keep asking. So remember our history of disease. This goes way, way back. We had miasma. What was miasma again? Bad air, yeah. It was this idea of a dark cloud hovering over us causing disease. So we have miasma. We also then found out about animalcules, the little beasties that were in pond water. And then over time, in the 1860s, we were able to convince people that it wasn't miasma. It was actually caused by germ theory. So this whole new idea came out that we had these invisible microbes that could make us sick. And again, that was a huge leap of faith. I mean, to tell people you're getting sick because of an invisible microbe, that's like telling people they're getting sick because of a gremlin. I mean, something you can't see. But it was a huge leap of faith, and they had enough data behind them to cover it. So nowadays, we have a whole group of doctors, scientists, microbiologists that study infection, that study diseases. So we have pathologists. Their job is to study the progression of disease. So from the beginning, the middle, and the end. They are trained to recognize disease, to diagnose people. We threw pathologists a loop about 15 years ago, probably closer to between 10 and 15, because measles came back. We eradicated measles, and all of a sudden in the ER they had kids coming in with measles, and the pathologists didn't recognize it. The pathologists didn't recognize it. And they were kind of like going back to their old medical textbooks trying to figure out, well, what are these signs and symptoms? They kind of ring a bell, I don't know what it is. And when they looked it up, they realized it was measles. It was so old school that we had forgotten. So pathology, again, is the study of a disease. A disease is any time your body is not functioning normally. So, for example, I have asthma, so technically I have a disease. Now, it's not something I can spread, it's something that's just mine, but it's still considered an abnormal disease because it's something where my lungs don't function the way that they should. My little alveoli don't work for me. You know, how frustrating is that? So that's the basis of asthma. I can also study the cause of the disease, and that's etiology. And the reason for that is because when Koch was first studying bacteria, he caused the ones that cause disease the etiological agent. That means the basis of the disease. So now they're etiologists. I like etiologists because it sounds like you're eating, but whatever. Okay. And then infection, which is where we're colonized by our pathogens. Now, the CDC will publish a morbidity and mortality report. I kind of mentioned that a few minutes ago. And this is where the CDC gathers data nationwide, and then they also work with other facilities in other countries to gather data. And they put together reports of the numbers of diseases, that's our morbidity, and how many diseases we have. And then they put together the number of deaths, that's our mortality. So they have this constant flow chart going, and every week they enter in more data. Now, in the beginning with COVID, we're still unclear what our numbers were. We finally gave up on that data. And the reason for that is we learned that we were often double and triple counting people. And so, for example, if people went in and had a COVID test, let's say they had one of the brain tickler tests, okay, and it came back positive, they were then asked to go in and get a PCR test in a few days to see if it cleared, and they did, and they counted that as 2 positives. Same person, 2 positives. And then there was also another time about halfway in where we ran out of testing material, so we're trying to be careful with testing material. So if somebody in a home became positive, they counted everybody in that home as positive. They wouldn't test you because they were running low on tests. So for example, if I had a mother become positive, then they would say, okay, well, your kids are by nature, since they're in your home, positive, your husband's positive, or your wife's positive, you know, everybody who's in your home is positive. We don't want to test because we don't want to waste the test. So again, that went from 1 case to maybe 4 or 5. So we kind of just gave up on the numbers. We're doing better now, but we kind of learned that we had a lot of double, triple counts, quadruple counts. We're still kind of working on that. Moving forward, we're much more careful about where our data is coming from because it was hard to keep track of all of that. That's also why our numbers looked so horrible in the beginning, and they've kind of backed off a little. We've gotten rid of a lot of our double counts. The other thing with epidemiologists is they try to figure out what the prevalence of a disease is. That's the percentage of the population that has the disease. That's kind of like our morbidity. Maybe 50% of our population is sick. Maybe 20% is sick. And that can be for a region of the U.S. This is where we see a lot of times in the news where they'll say California numbers versus Florida numbers, right? So they're comparing 2 different regions of the nation, and it's a percentage of who has the illness. And then one of the things that has really hurt us with COVID is the idea of a carrier. A carrier is somebody who has a disease, who is passing the disease, but has no signs or symptoms. This was what happened with our friend Typhoid Mary. So again, she had no signs or symptoms of the illness, but was passing it to everybody, and they were dying of typhoid fever. We run into the same thing with COVID now. People can have absolutely no signs or symptoms. That's why that screening check, I appreciate that they do it, but there's not a lot of data there because all they're asking you is, do you feel okay? And I'm glad they're doing that. That's good. They've actually had they were kind of giving us an update. They said they had a bunch of students recently. They were coming to campus and they're coughing, sneezing, and then they got up to the cart and they're like, stop coughing, stop sneezing. They're like, we're okay. They're like trying to not cough it. And they're like, no, we saw you coughing all the way over here. Like, y'all need to go home. So it is good. We're using it as a screening tool, but it's just one of those things where it can only work so far. One of the things you run into with COVID is, again, you might not have any symptoms. I told you about my friend, all she had was the smell. That was it. She felt fine, just a loss of smell. So one of the things that helps us with illness is, of course, that normal microbiota that we've been talking about, our normal flora. The fact that they're present, they're colonizing us, they keep a lot of harmful bacteria away and a lot of harmful other organisms away. We also have some bacteria referred to as transient microbiota. This is where we pick something up, we might have it for a few days and then it goes away again. So maybe I touch a door handle and I pick up some Staph aureus on my hand. The Staph epi is going to get rid of it, but for a few days I might have Staph aureus on my hand. You know, so I have a transient for a few days. Or maybe I have something where I get exposed to a fungal infection. So now I have a transient fungus living on me. It might not be enough to cause a disease, but it's kind of hanging around on the skin looking for a breach in my defense. And if it can't find one, eventually it'll die off. So transients can kind of come and go. A lot of times our normal flora is going to keep us safe because they have what's called microbial antagonism. And it just means that what they're doing is they're taking all the nutrients and space from anything that's trying to come in. And so their very presence protects us. We mentioned C. diff and how Clostridium difficile can, infect the intestine and cause really horrible illnesses. But as long as you have normal flora in your gut, you're fine. C. diff can't take over. It's only if you go on antibiotics for too long and lose your normal flora, the C. diff can come in and take over. So our normal flora really keeps us safe. So they occupy areas that pathogens might want to occupy. A lot of our normal flora produce acids that are able to kill off invaders. They also will produce bacteriocyanins or bacteriocins, which are basically like little death enzymes. But again, our normal flora, even though it seems like it's always a good guy, sometimes they can be opportunistic pathogens. So we mentioned E. coli in the wrong place can cause a UTI. So even though they're on our side, if they get into a place where they shouldn't be, they can cause an infection. Luckily, we've learned a lot about our normal flora, and we're still learning a lot. We're finding that gut flora can influence things like not only how we feel, but it can influence our mood. It can influence our stress levels. We're learning a lot about the gut. And one of the things that we are really pushing nowadays is when people take antibiotics to take probiotics. So you can take just a chewy or a pill, and it replaces the good bacteria that are being killed off by the antibiotic you're taking. And so it helps your body stay in balance. So that's always a good thing to do if you're ever on any kind of antibiotic. So let's do a quick understanding check. Can anybody tell me the difference between normal and transient microbiota? Excellent. Yeah, one of them is permanent and the other one is not. So one is there all the time. That would be our normal flora. And the transient is not. It comes and goes. How can opportunistic pathogens cause infections? What do? Did they suddenly go on the dark side? Yeah, by moving somewhere they shouldn't be. Exactly. So in other words, if they're in one location of our body, we're safe. If they go somewhere they shouldn't be, then they can cause an infection. They will take advantage. So when we're studying organisms, one of the things we actually still use to this day are Koch's postulates from the 1800s, which is pretty amazing that we can still use his work. Remember, dusting off the cobwebs, Koch's postulates were where he was able to prove that a specific disease was caused by a specific bacterium. So he would have a mouse that had a disease, he would isolate it and look at it, colonize it, and then inject it into a healthy mouse, and then confirm that that mouse ended up with the exact same disease and the exact same pathogen. And his postulates work so well, like I said, they're still in principle and still applied today. But we run into some issues with his postulates. And some issues that we run into are the fact that not all organisms always look the same. So Streptococcus pyogenes, for example, it can cause a variety of infections depending on where it's located and also depending on the patient's immune system. So strep pyogenes is most often associated with strep throat. But if strep pyogenes gets into the blood, it can cause toxic shock syndrome. What do we usually associate with TSS? Tampons, yeah. So what's the deal with tampons? If you have tampons inside of you, you have blood product inside of you. And if you don't change the tampon often enough, which is why the risk is highest for the super or higher absorbency, the blood is staying inside your body for longer. And if there's bacteria on it, it gives them a nourishing advantage. So now we have a situation where bacteria is replicating every 20 minutes. The tampon's not being removed for several hours. And so what happens is we get a chance to have an infection go into our bloodstream. We can go septic. That's TSS. So that's what the risk is there. It's not that your tampons are dirty. It's just that it's a risk with the bacterium if it's present. Strep throat is, like I said, the most common, but you can also get a skin infection. Has anybody ever heard of impetigo? Yeah, it's like a little rash around the mouth or on the face. And it's a surface skin infection where the strep will overgrow your staph epi. And
Is that different than eczema?
it is different than eczema, yes. Eczema tends to be more of an immune system issue. So eczema is where our body produces too much keratin. And so what happens is our skin gets too thick and it builds up, and then when it tries to slough off, it looks scaly or red or irritated because we have too much skin in that area. It's like sloughing off.
Is that the same for like eating when it's antibiotics?
Yes, where we respond to a bacteria, we build up the scar tissue too big. Yes, same idea. Our immune system is overreacting. Exactly. And our immune systems tend to overreact. They're wonderful. They try to protect us. But sometimes it's like I mentioned, walking through a spider web. They go, "Ooh," and they kind of like panic. And that's actually one of the things that we see with a lot of our COVID patients. We find that COVID patients that are on ventilators, if you do a COVID test on them, their viral counts are so low, they're almost not even registering. And yet they can't breathe still. So the question became, what is causing this? And it wasn't the COVID at that point. What it was lung inflammation. Our bodies were overreacting. And so they were causing inflammation in the lungs. We had more white blood cells moving there, more fluid buildup. And so a lot of times the way they treat patients that are in that situation is they will actually put them on steroids, which shuts down the immune system. It's like we have to reset it. We have to turn the immune system off, let it reset and calm down, and then let it kick in again. And how weird is that? It's like the way you live is to turn off your immune system, but it's a temporary thing while everything kind of resets. Because we almost get a positive feedback loop. You know, positive feedback loops are where we start going and we just keep going, and we keep going, that's what's happening with a lot of our immune systems. So what's happening here with impetigo is that we end up getting strep on our face, and it ends up getting an advantage over staph epi. It's growing faster, and it starts causing rashes or outbreaks. And it's a very itchy, painful rash for kids. We usually see it in kids. Adults, usually our immune systems and our normal flora are more established, so we don't see it as much. So it can cause a reaction. It has to be treated, but it's very different from strep throat. Cellulitis. Have you ever heard of cellulitis?
Yes.
Yeah, that can also be associated with staph. What is cellulitis? Anybody know? Yeah, it's inflammation. Is that what you're going to say too? Yeah, inflammation with the skin. So if you've had a wound or if you've had surgery or something done, if you look at the skin, if it's red, inflamed, swollen, it's having edema or swelling in the area, that can be a sign of cellulitis. And so we have a bacterial infection in the tissue. Strep causes this, staph causes this. So we have a bunch of organisms that do. And then also scarlet fever, that's the one I mentioned last time where you get the strawberry tongue, you can get a high fever, and you can end up getting really sick. And we see that when the bacteria is lysogenic. What did lysogenic mean again? It has something inside of it. A virus. Excellent. So lysogenic means the virus is inside of the bacterium. Okay, so when strep gets a virus, it can get really mean and cause scarlet fever. So all of these diseases are caused by the exact same pathogen. So it's really hard to try to pinpoint because, you know, in Koch's study, he said one pathogen, one disease. But here we have one pathogen and we have what? 2, 4, 5. We have 5 diseases, right? And that's just ones that I have up on the board. I'm sure they can cause more. They also can cause myocarditis, swelling of the heart. There's all sorts of things they can cause. Rheumatic fever, that's another one that strep can cause. So there's lots of things. So the other issue we run into with Koch's postulates is some diseases can only be caused in humans. Meaning that if I take strep throat, for example, and I put that same organism in a mouse, that mouse might not show strep throat. It might show something else. It might show cellulitis. So it's hard to get a correlation all the time. So we've kind of realized that there's some limits with what we can do with Koch. Overall, they're pretty good, but we always have to kind of keep in mind the limitations that we have. Now, when it comes to trying to classify illnesses, one of the things that doctors, physicians, epidemiologists will do is they'll study your signs and your symptoms. A symptom is going to be something that is basically related to us by a patient verbally. It's something that they feel, but it's not something that we can measure. So it can be, for example, a change in body function, like you're feeling tired, you're feeling achy, you're feeling worn out. So it can be a change in body function. A lot of times we'll ask you, for example, pain levels. Pain is relative too. We'll ask you on a scale of 1 to 10, what is your pain level? And you might have a dramatic patient like my child, my oldest daughter, who will tell you she's at a 30. And it's like, I don't think you're at a 30. No, I think I'm dying. It's like, no, I think you're fine. And then you have somebody like me, I have high pain tolerance. And so after I have surgery, I had a C-section, they're like, how are you doing? I was like, I'm good. I was up walking around. They're like, you shouldn't be walking yet. I'm like, no, I need to walk. I need to get up, So it just depends, right? So we try to measure on scales of 1 to 10, but again, it's very subjective. It's how you feel, Meanwhile, my child's laying on the ground dying again. A sign is easier because it's something we can measure. A sign is something like a fever. A sign is something like, respiratory distress, inflammation. It's much easier for me to gauge that because I have a number or a measurement that I can use. I can use a spirometer to see how people are breathing. I can use a temperature gauge to see what their temperature's at, a thermometer. I can see pus that could indicate an infection. So I look for all of this stuff. A syndrome is when I put my signs and my symptoms together. So I have, for example, if I said, what do you think of when you picture the flu? What are things that come to mind? Fever. What was the other one? Yeah, loss of appetite. Any others? Shakes, chills, body aches. Yeah, those are all like fevers. So those together are signs and symptoms. And that would be your flu syndrome. It's like we put it all together. So you feel a chill. I can't really measure it. I might see goosebumps, but you can tell me how chilled you are. So things like that. So again, another syndrome then would be something like AIDS. It would be something that we can measure and then also how the patient's feeling. So that can also be a syndrome. It's a grouping of what we kind of picture as characteristics of that disease. Now, the other thing about diseases is some are communicable. And what that means is they're spread from one host to another. So if I'm a communicable disease, then I can give it to somebody else. So COVID, for example, is a great communicable disease. Some diseases are contagious, and this is what COVID is as well. It is easily spread, right? Now, we've seen variations with how easily COVID is spread, though. We've had people who have had COVID who haven't spread it at all, and then we have super spreaders. You guys heard of the super spreaders. So what's the deal with that? Why is it that some people don't spread it and other people do? It has to do with a viral count or viral load. So if I have somebody that has like 100 to 1,000 viral particles, that tends to be low enough that they don't spread. If I have somebody whose body is carrying 10,000 particles, that tends to be high enough that they do spread. This is why there's this huge debate right now about kids. So there was a big, and then there's still a divide right now between the CDC and pediatricians. Because pediatricians are saying children do not spread COVID. Have you guys seen those studies? They've released a bunch of studies. And the reason they're saying that is because children aged 10 and under, they're too small. Their viral loads aren't getting big enough. Now, we classify children up to the age of 18. I don't know about y'all, but if you look at kids, they're very different size between 10 and 18, right? 18 is like an adult, basically. So that line of children, it's kind of being inflated. It's like, yeah, if you're over 12, you have a greater chance. Because some of our 12-year-olds are like the same height as us, right? So they can have an adult viral load. They could potentially spread the virus. So that's why there's this big debate. Is CDC is saying children are spreading, and the pediatric group is saying no, they're not. So there's a big debate there, and it's all about viral loads. How many viruses we carry as to whether or not we spread. So communicable means the disease can be spread. Contagious means the disease can be spread easily. If I get exposed to this person, I'm probably going to walk away with it. Okay? Sorry. And then a non-communicable disease is something where the person has the illness, but they can't spread it. Can you think of a non-communicable disease? Something somebody has and they can't spread it. I mentioned mine earlier. Asthma. Yeah, asthma is non-communicable. Another one's diabetes. Diabetes is non-communicable. So they have something, but they can't give it to you. A UTI would be non-communicable. It's not like they can give it to you. Okay. I'll go ahead and stop there, and we'll pick up on our next slide next time with our diseases in the population. Okay, all right, here we go. Okay, so chromogens, we have two different types of plates in front of us. We have bacteria on our urinary tract infection plates, UTI, and we have Candida, which is our yeast, in front of us. So let's start with our bacteria. So when I'm using chromogens, remember I'm testing to see what kind of enzymes the bacteria are producing and what kind of sugar they prefer to eat primarily, okay? Now, some of them are going to be a single color. Maybe the chromogen I have in there is pink for glucose. And then some of them like glucose and sucrose. So maybe sucrose is blue and glucose is pink. So if they're eating both together, then their color would be purple. So I see combos of colors. So that's why we see some combinations in there. So we're still testing for enzymes. And because the enzyme levels that are being produced can be really low, these tests are actually better than our pH shifts. Before we were using pH shift, but to do a pH shift, I have to have an enzyme breaking a substrate down enough to cause the pH to change. And then that pH change causes my pH indicator to change. But if I have an organism just eating like a little bit of glucose, maybe like 10 molecules of glucose, that might not be enough to cause a pH shift. We saw that in here. Remember sometimes we saw broth that was orange instead of going all the way to yellow? It wasn't producing enough enzyme. Chromogens are more sensitive. As soon as you produce it, boom, you get a color reaction. It's just like if you lick a couple of licks on a popsicle, your tongue turns color. Okay, so it's the same idea. So let's start with looking at our colors. What color is our pretty E. coli?
Purple.
Yeah, is it kind of like a purpley pink? Let me see, it should be kind of like a more pinkish color, but yeah, okay, so like a pinkish purple. Okay, so we'll put on here pinkish purple. I like that. And then what color is our E. faecalis?
Like blue purple.
Yeah, or like a turquoise. Do you guys think turquoise is good? Teal, I like that, like teal. I don't you feel like an interior decorator? It's like this is like a cornflower blue, and this is a teal. It's like okay, yeah, so lipstick. Okay. How about our citrobacter? What color did it turn? Yeah, denim blue. I love that. Yeah, we call that a navy blue, but like a denim blue. I like that too. Same idea. How about Staph aureus? Yellow. Yeah, kind of a yellow color or a yellow white, right? So like a yellow white. And then how about Staph sacro? Yeah, more like a cotton candy pink, right? It should be like light pink. Okay, so cotton candy pink. Okay, so looking at these, do they all look different? Yeah, and that's the key. Is if you have this plate in front of you, you don't have to have any type of microbiology background, then you can identify what organisms you have just based on their colony color. So these things are great. When I used to work in a micro lab, if I had somebody coming in that was new, one of the things they would do is a bunch of gram stains and they would read chromogen plates because you really can't mess it up. I mean, it's just, we would have a colored chart right beside their plate. They put their plate on a chart and they would just compare. The only time it can mess up, I had a few lab techs that came in, I would give them the color chart and I'd look over and they'd be kind of like doing this. And I look at them, I'd be like, is something wrong? They're like, and they're trying to struggle through it. And I realized they were colorblind. Yes, there are a few lab techs out there that are colorblind. And so they were like seeing shades of gray. And that's like all they were seeing. So yeah, and great text, but it's kind of funny they didn't want to say anything. They're looking at these colors going, I'm just not seeing it. Yeah, so sometimes you can have issues with that. Did you have a question?
No, I was just
yeah, okay, great. Okay, so our Candida plates, we have to let Candidas grow a little bit longer. In your lab book, it said you need to let them grow for 48 hours. The reason are that Candidas, remember, are eukaryotes. They grow slower than bacteria. Bacteria are every 20 seconds, or sorry, every 20 minutes. Gosh, I'm glad it's not 20 seconds. Every 20 minutes. For a eukaryote, they take a good day or so, to start getting cell division. So they're just slower, and we need to give them a full 48 for their time development. So let's look at these guys. For C. albicans, do you see a pretty green color? Yeah, you see pretty green. For C. krusei, what do you see? Yeah, like a greenish blue. Okay. And then what do you see for tropicalis? Purple. Yeah, so we usually see like a purple.
Oh, wait. No, for the second.
For krusei, you should see pink. Are you seeing more purple? Yeah, pinkish purple. Okay, that's okay. Yeah, pinkish purple. I was kind of going with the greenish because I wasn't sure if it converted since we haven't seen them. They've been sitting in the fridge for so long. So it is still more of a pinkish?
Yeah, pink purple.
Okay, pink purple. Okay, so pink purple. And then tropicalis should be blue, but like a bluish purple. Is that what we're seeing? Because they've been sitting a little longer than normal. At 48 hours, these things, these are blue, these are pink, and these are green. Hold on, let me Yeah, so this right here is albicans, and this is tropicalis, so it's dark blue around the edges. The reason it's more white here is because they're running out of food. Where the heaviest growth is, you see they're kind of white, they're running out of food. When their heaviest growth is. That's why they have kind of that weird variation. So again, you can tell a difference between the two, right? Or between the three, I should say, right?
So it would be purple if you saw it sooner?
Yes. It would be, the tropicalis would actually be more blue if you saw it sooner. The krusei would be more pink if you saw it sooner, and the albicans is always green.
Oh, no. It was mixed up.
Yeah. Now remember, we talked about an emerging infectious disease called Candida auris. Candida auris looks just like albicans. And that's one of the things that has made it very difficult to diagnose and treat is if you set up a patient sample and they have auris, it gets misdiagnosed as albicans 9 times out of 10. The difference is auris is resistant to almost everything out there. So they start to treat it like an albicans and it doesn't go away. The other problem is that the auris is highly contagious and albicans isn't. Which means if you have a partner or a roommate in the hospital, there's a chance they can encounter it. So they're trying to figure out right now, they're trying to develop a chromogen test to tell the difference between these two. So far, they haven't been able to because their biochemistry is so similar. So we're still kind of on hold with them. Okay, one other thing I want you to do today before you leave, we're going to set Hands, but just before you leave, is don't forget about your winograd seeds. I'm going to unlock the garden area, and just on your own, you can walk by and look and see how our bottles are developing. So remember, we have our anaerobes at the bottom, our aerobes at the top, and we should be getting a nice color change with them. So today we'll go out there, we'll look at them, we'll see how their color is developing, and then next time we'll kind of look at it again, and I'll start doing more of a chart about them, okay, to make sure that we're understanding what's going on. But remember, they're basically little ecosystems. We want to see how our little ecosystem is developing. Look for any color. Do you see any color yet in any of these? So that's what you're going to do today. The easiest way to do winogradzkies is to take a picture with your phone, and then you can look at them at home and write in your notes. That's honestly the easiest way. But I'll unlock the garden as soon as I get done talking to you. Okay, so the lab we're going to be doing today is going to be called the Miller-Hinton lab. And this is going to be used for antibiotic testing. So we mentioned before in the lecture that this lab is or that this type of testing requires us to be able to use a certain type of media. Were we able to use just regular compound media or just general media, or do we have to use chemically defined? Yeah, it has to be chemically defined. It can't be complex. It can't be regular. I have to know every ingredient that's in my Miller-Hinton, because the only variation is going to be the antibiotic. So the way I'm going to set these up today is you should have 2 plates in front of you. One of you is going to set up with E. coli, and one of you is going to set up with staph. Be sure to type cap and invert because they're all sleepy in there, okay? What you're going to do then is with your setup, you don't even have to use a loop today. We're going to use a, should I have to use a loop? Don't want to have to use loop. Now I have to think about it. Don't want to have to use. Now I'm going to have to use the swab. Sorry. I'm going to have to use swabs. More of my swabs. Okay, I'll get some more swabs. So we'll have us use a swab. And the easiest way to do this, and I'll get us some more swabs because we're low up here, is you're going to invert your bacteria to wake it up. Okay. Then you're going to dip your swab into the bacterium. Get it nice and coated with bacteria. When I pull it up, I'm going to kind of gently push it to the side. I'm not really wringing it out, but just enough to where if I carry it, it's not dripping on me, okay? And then the way I'm going to inoculate a Mueller-Hinton is I'm going to make a lawn. That means a full growth of bacteria. So I hold the plate like this. I don't want to hold it like this because it's getting contamination. I hold it like this so the air currents don't fall in it. And what I'm going to do is I'm going to start at the top and I'm going to swab gently back and forth, back and forth, back and forth, making sure I'm overlapping all the way to the bottom. Then I'm going to do a turn. And now I'm going to start at the top. I'm going to turn my swab a little and do the same thing back and forth so I'm overlapping. So what I see on here is I see stripes going this way, and now I have stripes going this way. Okay? I'm going to turn one more quarter turn and do the same thing. I'm making sure I'm getting this nice lawn. And then the very last step I'm going to do is I take the swab and roll around the edges. That way I make sure that all of the bacteria are covered. Did you guys see that over here? Sorry, was that angle okay? So I swab back and forth. I do a quarter turn, swab back and forth, and then at the end I do a wrap around the edges. Okay? I let my plate sit for a few seconds. This can go in my biohazard, where my plates go. Okay, so in the biohazard bin. I let the plate sit for about a minute or two while it dries and absorbs into the surface. And then I'm going to add antibiotic discs. So at your station, you have 4 types of antibiotic discs. And these can be a little tricky to get out. What you're going to do is you're going to use your tweezers. You're going to gently kind of push on the side of the disc, and I'll probably shoot one out of here. They kind of shoot out fast. But you can kind of push it to the side. You grab the antibiotic disc on the side, and then you're going to put it down on the surface of the media. So let me show you how that looks. So I have it in my tweezers. I'm going to gently push it onto the media. So it's sitting like this, and then I take my tweezers, and I just gently tap it to make sure that it's sticky. Okay, I'm not jamming it in there. It's sitting right on top. So it's very gentle when I put it on there. But check it out. It absorbs some moisture, so if I turn it upside down, it's not going anywhere. All right? Then what I'm going to do is I'm going to put these plates into the incubator, and I'm going to put them in like normal, lid side down, but I have the disc sticking on there. It's not going to fall. And then next time we're going to come in and we're going to see how our zones of inhibition are doing. We're going to see how this antibiotic disc diffused out and whether or not it's affecting the bacteria. All right? So that's what we're going to be doing today. Does everybody know what we're doing? All right, I'm going to get us some more swabs. So in the meantime, you can go ahead and throw away your chromogen plates, and I'll get you some more swabs and get started here.